Hi, my name is Latisha Franklin, and this is my lightning talk or my informational talk. So as I just said, my name is Latisha. I am a 7th-year PhD candidate in biochemistry, microbiology, and molecular biology program here at Penn State University. I'm originally from Mobile, Alabama, which, if you did not know, is the birthplace of Mardi Gras, not New Orleans, Louisiana. Also, I work with C. elegans or microscopic worms. I have inserted a picture here of the worms. They're zoomed in. They are important because most of the pathways and things are conserved in them. And so we can use them to study and understand other organisms, such as us humans. I'm also a homeowner. I won awards for presentations. I was on the homecoming court in 2020. And I'm also a business owner, which is called Gratified Grad. So my research is on understanding muscle dysfunction that's associated with inborn errors in purine metabolism. Purine metabolism is important for two main reasons. The first being that it generates the adenine and guanine, which are the backbones of our DNA, but also it helps to maintain our energy storage and the energy that we need to consume. Adenylosuccinate Lyase Deficiency, or ADSL deficiency, is at the intersection of genetic, metabolic, and nervous system diseases or disorders. It is an inborn error in purine metabolism. We'll get to that in a moment. And it is a recessive genetic disorder, meaning both parents have to have a mutation in order for you to see a phenotype. And so some of the things that we notice in people who have adenylosuccinate lyase deficiency is misshaping or abnormal facial structures, sometimes the absence of speech. This disorder is often misdiagnosed, therefore it's underdiagnosed, because the only way to actually confirm that a person has this disorder is through measuring metabolites and looking for specific metabolites in either urine, cerebrospinal fluid, or the blood. And so a lot of people are often misdiagnosed as having autism because the older you get, or the older one would get with this disorder, the milder the effects are. Because if a person has a severe case of ADSL, they often do not make it out of infancy due to the seizures and the brain development concerns. And so, as I said, C. elegans are a great model organism to use because it is conserved across humans to the animals. And so if you notice, a lot of the enzymes that are in the human pathway for purine metabolism are also in the C. elegans pathway. The work that I work on and will be showing to whoever comes to work with me looks at some research started by 2 undergrads in the lab about 7 years ago. They started looking at this lack of ADSL, lack of this gene in C. elegans, as we've been talking about, and they saw that there is developmental delay, movement defects, sterility, and reduced size. And so you don't have ADSL, so you get an increase in SACARP, but you also have a block in de novo synthesis. Oh, and also I'm looking at the movement defects. I also look at reduced size. We go about looking at this taking a very genetic approach. We use RNAi, which just basically reduces the function of the enzyme, or we use a mutation, which completely removes the function of enzyme in totality. And then we do assays such as looking at behavior. We look at because the animals are transparent, we're able to actually see the organs or the internals of the animals. And so we take images of them, we do metabolomics where we measure metabolites. And yeah, we also do the behavior, like I said. So an example of the behavior is putting the animals in liquid. And so the animal's natural response to being in liquid is to swim. And so this is what a swimming animal looks like. The software is actually tracking the animal. That's why it appears green here. And so from that, we can actually take a couple of measurements. Here's thrashing. Basically, this is showing that ADSL mutants as well as ADSL knockdowns are both decreased in their thrashing ability. And then we can also measure the crawling speed of the animals. So that's an equivalent of walking in people. And we also see that the crawling speed is significantly decreased when you don't have ADSL. Another example that I mentioned is metabolomics, looking at the metabolome. So once again, we collect the animals, we extract all the metabolites out of the animals, and we run that sample through the LCMS instrument. From that, we get all of the metabolism data that we could ever want about the animals. And then we can go in and look at specific pathways and measure specific metabolites and make those connections to understand what is changing, what is causing the phenotypes, and what can we correct for. Thank you. And if you have any questions, feel free to reach out to me. My email address is lpf113@psu.edu. I hope you enjoy, and I look forward to working with some of you.